Hello all, in this video we'll see about transport across cell membrane and molecular motors. Transport. Why do we need a transport? Transport is essential in a body to transport ions, electrolytes and gases, etc. So types of transport. Types of transport, there are two types, passive and active transport. Passive transport. Here there is no energy expenditure and the molecules are transported from higher region to a lower concentration along the concentration gradient. Types of passive transport are simple diffusion, facilitated diffusion, and some special types. They are osmosis, bulk flow, and filtration. Now simple diffusion. Simple diffusion is movement of molecules from higher concentration to lower concentration. Lipid soluble substances, they directly diffuse through the cell membrane, like oxygen, carbon dioxide directly diffuse through the cell membrane, whereas lipid insoluble substances, they move across the cell membrane through gated channels like electrolytes. There are three types of gated channels: voltage gated, ligand gated, and mechanical gated channel. Voltage gated channel, they open when there is a potential across the gates, and ligand gated channels open when the ligand binds to the gate, and mechanical gated channels, they open when there's pressure applied on the gate. Now we'll see tonicity of a cell. A cell is said to be isotonic with plasma if the cell's concentration is equal to the plasma concentration, and hypotonic if the cell's concentration is less than the plasma concentration, and hypertonic if the cell's concentration is more than the plasma concentration. Osmosis. Osmosis is the movement of water molecules from a region of higher water content to a region of lower water content across a semi-permeable membrane. There are two types of osmosis, exosmosis and endosmosis. When water exits from a cell, it is called exosmosis. When water enters the cell, it is known as endosmosis. What is reverse osmosis? Reverse osmosis ensures water from a lower water content to a region of higher water content. Example, we get clean water from a salt water. You have to know about three types of pressure: osmotic pressure, reverse osmotic pressure, and oncotic pressure or colloid osmotic pressure. Osmotic pressure prevents the water entry from a hypertonic solution into a hypotonic solution, and reverse osmotic pressure ensures water from a hypertonic solution to a hypotonic solution. Oncotic pressure or colloid osmotic pressure. This is pressure exerted by the colloids like proteins. Bulk flow. This takes place in lung alveoli. Usually we know that diffusion occurs across the lung alveoli of gases. There is also bulk flow of exchange of gases of oxygen, carbon dioxide across the alveoli membrane, and the filtration process is seen in renal glomeruli. We call it as ultrafiltration. Now we'll see facilitated diffusion. There's difference between facilitated diffusion and active transport. The only thing common between them is the molecules are transferred across the cell membrane through a carrier protein. In facilitated diffusion, the molecules move from a higher concentration to lower concentration, and there's no energy expenditure, but it is through the carrier proteins. Now we'll see active transport. In this active transport, there is energy expenditure, and the molecules move from a lower concentration to a higher concentration. We divide them into primary, secondary, and special type of active transport. Before that, we'll see about carrier proteins. They are uniport and symport or antiport. Uniport means there is only one molecule transported in single direction. Symport and antiport, there are two molecules are transported, but in symport the molecules are transported in the same direction. In antiport, the two molecules are transported in the opposite direction. Now we'll see primary active transport. This requires direct ATP hydrolysis. We get energy through this, and the example is sodium potassium ATPase, which are present in all cells, and proton pump, which is present in gastric parietal cells. We'll take example of sodium potassium ATPase pump. In this carrier protein, intracellularly there are 3 sodium attached to it, and extracellularly there are 2 potassium attached to it. When the ATP is hydrolyzed, the energy released makes a conformational change in the carrier protein, and finally the ions are distributed to the cellular compartments. In secondary active transport, we use stored form of energy but not from the direct ATP hydrolysis. Example, sodium glucose co-transport and chloride bicarbonate counter-transport. This is called as Hamburger's phenomenon. A special type of active transport are 3 types. They are endocytosis, exocytosis, and transcytosis. In endocytosis, there are 3: pinocytosis, phagocytosis, and receptor-mediated endocytosis. Pinocytosis means cell drinking, where macromolecules like antigens and bacteria are taken into the cells, and finally the lysosome digests the macromolecules. Phagocytosis, also known as cell eating, where molecules larger than macromolecules like larger foreign bodies, they are digested by the lysosomes. Cells carrying out phagocytosis are monocytes, neutrophils, and tissue macrophages. Receptor mediated endocytosis, here receptor coated pit is present in the cell surface. To it, ligand would be bound and it would be engulfed into the cell and it would be metabolized. Examples are cholesterol metabolism and hormone metabolism takes place through receptor coated endocytosis. Next is transcytosis. In this, first receptor mediated endocytosis takes place, and finally exocytosis takes place. Example for transcytosis is HIV replication. Now exocytosis. Exocytosis, also known as cell vomiting. Here the substances are expelled out from the cell. Example of it is acetylcholine release from the nerve terminals. Now we'll see about molecular motors. These are motor proteins which help in cellular movements. There are 3 types of motor proteins. They are kinesin, dynein, and myosin. And kinesin and dynein are microtubule based, and myosin is microfilament based. Microtubule contains a positive end and a negative end, and kinesin moves over the microtubule towards the positive end. This is known as anterograde transport, and dynein moves over the microtubule towards the negative end. This is called as retrograde transport, and myosin slides over the microfilament. All 3 proteins have a head and a tail. The head contains ATPase enzyme which breaks the ATP and produces energy which helps in motor functions of the cell, and the tail contains a cargo, that is a substance which has to be transported. Common functions of these proteins are transport of synaptic vesicles from the cell body to the synaptic terminal, and cell division by spindle formation, and also muscle contraction. That's about cell physiology. Thank you.